Hi, everyone. Thanks for joining our webinar today. My name is Brent Cunningham from HORIBA, and I'm your facilitator. HORIBA and PureNanoTech are proud sponsors of the Nanobubbles 2024 conference. It's a conference where researchers from around the world come together to discuss the recent advances and applications of bubbles. This is part one of a three-part series discussing nanobubbles. With me are our guests Dr. Hua of PureNanoTech and Julie Chen, Particle Science Liaison from HORIBA, who will talk briefly about methods for characterizing nanobubbles. But with that, it is my great pleasure to introduce you to our speaker, Dr. Hua. Dr. Hua is the Chief Technology Officer of PureNanoTech, where he leads the technical development and R&D focusing on product development and demonstration projects. With his extensive background leading significant projects with the USDA, USGS, NJDEP, and the EPA, Dr. Hua brings a wealth of knowledge to the table, and in this webinar, he will dive into the exciting world of nanobubble technology, sharing insights that open up the nano world to us all. Dr. Hua, with that, we're all very excited. I'll pass the ball to you, so whenever you're ready, please share your screen.
Thank you, Brent. So before we further go to the nanobubble, I'll just quickly introduce our company, the PureNanoTech. We are a company that provides cutting-edge nanobubble generator equipment and services. We're committed to offer solutions that are not just innovative, but also stable, efficient, eco-friendly, and tailored to meet every needs to our clients. And besides, we offer viable carbon reduction solutions in this process. We have a diverse team of experts, each bringing wealthy experience in science, technology, and business, so that will ensure we deliver the solution to meet the expectation to our clients. And yeah, I'm Likun Hua, and I'm the Chief Technology Officer. I take care of the R&D, the tutorial of product and tech support. So if you ever bought a product from PureNanoTech, you probably know me. And without further ado, let's go to today's webinar. I'll give a brief introduction about what the nanobubble is and the application and the potential of nanobubbles. I'll then talk about how nanobubbles are generated and a few case studies. Then our partner Julie from HORIBA will talk about HORIBA's nanoparticle tracking analysis technology. This is the one we use to detect and analyze our nanobubbles. And of course, it can do a lot more different nanoparticles tracking applications. Let's leave that to Julie. And yeah, at last, we will do a little Q&A session, and that's about it. So first, what is nanobubble? When we mention about bubbles, such as air bubble in water, we know that the air bubble in the water will go up, flow to the surface, and eventually burst at the surface of water. However, the nanobubbles does not work that way. The nanobubbles are very small. The diameter is less than 1 micron. So it brings it a excellent stability because their sides are too small, so the buoyant force cannot do much on them. Also because there are low mass in the bubble, so gravitational force also have minimal effect on them. So basically, they are suspended in the water. This also gives the nanobubble a high mass transfer efficiency. Imagine if you want to transfer bubbles in the water, it will stay there. It will not go up and go out. So without the buoyant force and the gravitational force, what makes more roles in nanobubbles are the Brownian motion, the surface charge, and the surface tension. This gives the nanobubble the feature of hydrophobicity, the negative charge, and even hydroxide radical if we're using the right gas. With all those properties and features, we consider the nanobubble is a platform technology. That means different properties and features can go to different applications. For example, the enhanced mass transfer can go to the aquaculture. If you want to bubble your pond with the gas to grow, to raise fish in there. And the negative charge, because it can attract nutrients on the nanobubble surface, that goes to the aquaculture, the agriculture application to help the plants absorb those nutrients better. And the ROS formation, this hydroxide radical can help to do the disinfection, so surface disinfection, surface cleaning. And at last, the hydrophobicity can overall reduce the surface fouling problems. So as you can see, we try to cover a lot of industrial areas with the nanobubble technology. Some of them we already deeply involved, such as agriculture and surface cleaning. Some of them we are engaging them, such as aquaculture, the healthcare, and lake pond restoration. And some of them we are still exploring, such as oil and mining. So we really open to the collaboration with our researchers and industrial partners. Before we further go to the case study of in different industrial sectors, I want to talk a little about how the nanobubbles are usually generated. There are several ways available now in the market. The first one is called the Venturi method. This one is actually using high pressure gas and high pressure liquid, force them to go through the Venturi tube and then create nanobubbles in this process. The second one is called the throw method. This one is actually using the gas, inject the gas into a spiral liquid flow and use the shear force to shear the bubbles to a small one, which creates the nanobubbles. The third one is a little bit different, called ultrasonication method. This one does not carry any gas, so basically what's inside of the bubble, we call it cativation. There's nothing there. This one is only suitable for surface cleaning. And the last one is a membrane method. This is actually what our company are using. This is a spin-off technology from New Jersey Institute of Technology. And with this membrane method, we first go to the agriculture sector, which is what we involved most deeply. And in the agriculture sector, what nanobubble can do is first, we enhance the farming efficiency and conservation. The efficiency we enhanced is the crop yield and the operational efficiency. And what conserves here is water, fertilizer, and even chemical pesticide usage. We also try to use this technology to solve the food desert community problem, improve their access to nutrient food, and encourage those communities to engage in cost-effective and healthy farming practice. And like I mentioned before, this technology can also reduce the carbon footprint. And so we are reducing water usage and fertilizer there. So yeah, let's dive into the case study we have here. This is the one, the picture shows here is the one we tried in, we call it hydroponic agriculture, but we also do in traditional farming in soil. As you can see in the picture here, the plants with our nanobubble water irrigation grows better, larger than the one with the normal water. The plants used here are parsley, basil, and cilantro. Also, you can see the root are also growing better with the nanobubble water irrigation. This is what you can see in the plant growth improvement. What you cannot see also includes the like the water quality, because the biofilm is reduced in the pipes of the irrigation, and in traditional farming, we also can make the soil better, improve the beneficial microbes activity, such as or lower the water surface tension to promote the water infiltration. And since we're here, probably you are wondering how nanobubble can do this, how nanobubble can promote the plant growth. Well, basically, the major mechanism to enhance plant growth, we give them to four major ones. First, the nanobubble actually could increase the local oxygen level because we're using the oxygen nanobubble or air nanobubble in the agriculture application. So the oxygen level in the water or the soil system gets raised, and it will promote the aerobic respiration near the roots, since you saw the roots are growing better, and it leads the increased energy production and enhance the metabolic activity. The second one is a nanobubble can help the nutrient being delivered better because a nanobubble is a negative charge. It can absorb nutrients on the nanobubble surface and mobilize the nutrients in the soil, in traditional farming, and in the water, in the hydroponic farming. It facilitates the accessibility of nutrients to the plant roots. And this one will improve the nutrient delivery, lead to better absorption of those nutrients by plant roots, and promoting healthier and more vigorous growth. The third one is the reactive oxygen species, the ROS, and such as hydroxyl radicals. It will suppress the biofilm buildup in the irrigation system. You know that the nutrients in the irrigation system should go to the plants, not those biofilms. So to reduce those biofilms, that reduce the nutrient, absorption by those biofilm and make it more use for the plants. And the last one is support the soil microbial activities. So it actually supports the beneficial microbes and aiding the organic decomposition and the nutrient cycling. So this is what we are doing in the agricultural sector, but we're doing more, such as surface cleaning and sterilization sector. In this sector, we are also using oxygen nanobubbles. So the traditional method of cleaning, maybe you just use water to wash them or use the chemicals. Well, chemicals are not good. In some of these industries, they use ozone or even worse, the chlorine-based chemicals to wash or cleaning the surface. And this will cause environmental problems and chemical residuals. But in the nanobubble technology, you don't need to use much of the chemicals, or even you don't need to use chemicals at all to enhance the surface disinfection because of thanks to the reactive oxygen species and the oxygen itself, it can help to clean the surface of whatever you're applying to and disrupt the microbial biofilms, including E. coli and many of those bacteria in there. So we use them in the meat processing and fruit or other distillation removal sector. This is one of the case study we collaborated with a chemical company on disinfection demonstration. This one is 200 GPM capacity one. That means in 1 minute it can produce 200 gallon of nanobubble water, oxygenated nanobubble water. This is the largest single unit we have because we have a modularized design, so it is actually an easy scale up for higher capacity. And this project is ongoing. The next sector is a little bit interesting. It's called carbon capture and waste to resource. So we know that greenhouse gas play a big role in nowadays the global warming, and CO2 is the most recognized one of the greenhouse gas. Things, so hence we have this carbon capture concept. And this one we actually collaborated with New Jersey Institute of Technology to build up a microalgae photobioreactor. So we know that algae is actually live on photosynthesis, so it needs CO2 and light. The CO2 can come from different sources such as flue gas or heating facility. Anyway, these are waste gas to them. We use pipes to guide those gas into this photobioreactor to cultivate algae. And we know that the CO2 is soluble to water, but only in limited amount to an extent. With the nanobubble water, we can actually enhance the CO2 in the cultural medium or water, whatever it is, enrich this medium with CO2s. So it will rise the overall efficiency of the photobioreactor, make it eat more CO2 in plain language. And at last, those microalgae can be collected by us. Those are actually resources, important resources for personal care products, such as cream or face mask. And it can also be used as a biodiesel or biofuel. So there's a waste to resource. The last one case study shown here is also related to algae, but this time we are not using them, but get rid of them. So this is also a bad result from human activity, I think. This is harmful algal bloom in New Jersey lakes. We know that eutrophication and climate change can cause those harmful algal bloom. And back in 2019, there are several warnings was raised in New Jersey lakes for those recreational lakes because there are algal blooms in there. So those recreational lakes are no longer suitable for recreation in some season, summer season. So this one we collaborated with New Jersey Institute of Technology and Midland Environmental Research Institute. We try to use the nanobubble generator, build them on a boat to improve the water quality and the overall ecosystem. In this application, we use air nanobubbles. The nanobubbles actually help the algae to float and coagulate together. Once it's surfaced, we use equipment like skimmers and centrifuge to collect them. And the result in this project lakes are good. The restoration works are successful and removed a substantial number of odorous algae in this project lakes. And oh, by the way, there's no chemical reagent is needed in the whole process, so it eliminates the possibility of secondary pollution. So at last, I want to mention a little about our existing products. So we have two product lines. One is called the Kit series. It's the nanobubble core module without any pump or other equipment. Easy to carry, very small, and they are suitable for researchers. You can build your own reactors or your research equipment with this attached to different things, and these are modularized design, so you can choose different capacity. The second line is called generator series. They are modularized, integrated with different equipment inside. The standardized one have air compressors or water pumps in there, but we do take customization to our customers. So as you can see, we face to researchers and industry application. We are also very welcome, open to the collaboration from researchers and industry partners that explore more applications on nanobubbles. So finally, I want to show you the performance with our standardized equipment. So this one shows the nanobubble in the water concentration and the mean size of the nanobubble. But we can do customization if you want a different size. We can build it for your request. This data is acquired from HORIBA's equipment, the ViewSizer 3000, and I think Julie will elaborate more details about this equipment.
Thank you so much for the introduction. Again, this is part one of the 3 series. Let me go ahead and share my screen. So just as there are different ways of generating bubbles, there is a number of methods used to characterize them as well. Here I'm listing 8 different techniques. each takes a different approach, providing different distribution output, such as intensity, volume, or number. Number size distribution output is perhaps the easiest to understand. If you think about microscopy, you are taking pictures of the particles through the lens of the camera, and then you go in to count the number of particles seen one by one. So that's what it means when we say number size distribution. Volume means that you're assuming each particle is spherical and you're measuring its spherical volume. Intensity means that you are looking at how strongly that specific particle scatters a laser light source or some sort of light source. So they're quite different. In general, it is advantageous to choose one or two orthogonal technique to verify your result because it's very unlikely that two techniques would have the same bias. But in the case of characterizing nanobubbles, just based on the approximate limit of detection, you can see that DOS and NTA are the two clear winners here. So DOS can measure 5 nanometers and up, whereas NTA can measure 30 to 40 nanometers and up. So between these two, the question now is, okay, what is your required bubble concentration and what kind of output do you want to see? So the name of the game is to track illuminated particles. So you use one or two laser light source to illuminate any particles that's in a solution. Large particles are going to move slower, but scatter light stronger. And inversely, with the smaller particle, it's going to move faster, but it's going to scatter light weaker. So the analysis of the track is to use a camera to essentially look at the random motions of these nanoparticles. And then it's done by evaluating the mean square displacements of each one of this particle. Plug it into an Stokes-Einstein equation, and then you have a size distribution. Now, if we know the investigated volume, we can then use that information to give you concentration, such as particles per mil. So why do you need 3 lasers? What kind of sets the view size or NTA apart from the typical 1 laser NTA? And really answering the question why PNT, Pure Nanotech, use this specific instrument to characterize their nanoparticles. So I'm going to show a video. What you're seeing right now is a bunch of nanoparticles moving in Brownian motion. They randomly scatter light using different wavelengths. So with the view sizer, you have the blue, you have the green, and the red lasers. 3 lasers are turned on to illuminate particles simultaneously. And that's a patented technique developed by the University of California, San Diego. So here I'm going to pause the video. You can see that if you have large particles, whether it's large droplets, nanodroplets, or nanobubbles, red laser at 633 nanometers is going to scatter light and allow the camera to track in a very defined manner. Whereas if you use blue, then you're going to see kind of a useless blob that is not clearly defined. And as they move in Brownian motion, they may touch each other and they may crowd each other, and therefore the algorithm would not be able to decipher if this is 1 or 2 particle. But if you have a very small particle, for instance here, I'm going to pause again. If you have small particles such as really small ultra-fine bubbles down to the 30 to 40 nanometer size ranges, you will need a blue laser to illuminate those smaller particles. Looking at the red, you're missing out this 3 additional count. So that really does affect your concentration information, and from there, it would affect your total surface area, which is a parameter that's quite relevant in nanobubble application. So what the system does is it illuminates particles using 3 laser wavelengths. The algorithm, it takes the video, and then the algorithm will go in to analyze the video and decide for us which laser wavelength is most appropriate for that specific particle. So from there, we're not overcounting, but it gives us a very good size distribution, whether it's big or small or when they coexist together. Here we're looking at ultrafine bubbles, aka nanobubbles repeatability runs, where 5 replicates of 1 aliquot of the nanobubble sample submitted to our lab was measured. You can see that with this sample, the concentration is somewhere around 1.27 times 10 to the 7th, which is 2 decades lower in concentration compared to the one that PureNanoTech has generated. But regardless, it's a good example to show that nanobubbles are in nature polydispersed. When you measure enough particles, you can achieve a coefficient of variation of 2.46% for concentration and 1.94% for the median. Again, reproducibility is very important because what's the point of measuring if you cannot remeasure what you produced? So here is a study where we have 3 different analysts, really my colleagues in the lab. We all measure a aliquot of the sample that was submitted to us, and this is O2 nanobubbles. So analyst 1 measure 5 different times of the one aliquot of the nanobubbles. The other person measured 3, and the other person got tired, so measured 2. But if you compile all the data together, you can see that the reproducibility of this nanobubbles is quite good. So what it tells me is we're removing sampling issue, we're removing instrumental bias, and it's suggesting that this nanobubble is quite stable over time. So the question when it comes to nanobubble and NTA is always, how do I know my concentration is correct? How do I know that what I measure is what I have? Since NIST has not released a concentration standard, they have a size standard. We then kind of move towards third vendor, which is Nanocomposites, a company that sells various different reference material. Here we have 10 nanometer gold nanospheres. On the left-hand side, you see the result produced by the ViewSizer 3000, which is the NTA system that HORIBA offers. You can see the distribution is rather narrow. When you compare the data to TEM, it measured 99.4 nanometers, and particle concentration, which is measured using the ICP, single particle ICP-MS. You can see that in the next table, the ViewSizer measures size and concentration really well. TEM, the gold standard, the of sizing measures 99.4 with a maximum 1.5 and a minimum 93. NTA measures the hydrodynamic size diameter of 1.4. So it's well within the specification, and that same thing applies with the concentration. So this is how we usually know that the concentration is correct. You can also do a dilution series, but really what we're trying to say is when it comes to nanobubbles, it's advantageous to have more particles or more bubbles per mil, but it is important to know that you are measuring accurately. So in summary, ViewSizer 3000 is a patented technique that can offer simultaneous multi-laser nanoparticle track analysis. Each one of those lasers can adjust its own laser power independent from the other. And therefore, if you have smaller particles, you can increase a specific laser power to enhance the illumination. It uses a high resolution camera that allows you to look at mixtures or polydispersed nanobubbles. In addition to it, many users have commented how much they enjoy using a quartz cuvette-based cell assembly as opposed to syringe injection. That allows them to minimize cross-contamination, allows them to use various different solvents, whether it's oxygen and water, or maybe they are dispersing their nanobubbles in organic solvents. This is a type of a cell assembly that's completely compatible. So for more information, you can certainly reach out to us or leave us a quick comment. I'm going to go ahead and pass the ball back to Dr. Hua, and we will kind of open the floor up for Q&A session.
Yes, thank you, Dr. Hua and Julie. We do have a few questions that have rolled in so far. The first, with numerous nanobubble generation technologies, could you highlight the main differences between membrane methods and other techniques?
Okay, so yeah, there's some different, there's pros and cons for different technologies. For the membrane method, is actually the benefits of that. It does not need a very high pressure gas or high pressure liquid. It does not mean it cannot withstand high pressure gas and liquid, but does not need it. So during the process, the membrane process is actually does not need much of the power and power consumption, and you have less risk of pressure issue, the plumbing. This actually all reduce the cost during the operation and installation.
Okay, thank you. We do have some rolling in through the chat too. Someone was asking if you could elaborate the ROS production for insect pest management applications. If you can elaborate on that.
Yeah. So in that part is more focusing on the suppression of microbes during different applications such as surface cleaning or the aquaculture suppressing the biofuel. So the ROS is actually need the oxygen or air nanobubble to be used in the nanobubble generator. You cannot do that with other gases. So those are generated at the surface of the nanobubble itself, and also the nanobubble is negative charge. It will, only absorb along with those particles and go flow with the water, go to wherever you want to go. So yeah, if that's for the surface cleaning, that will control these pathogens on the surface, whatever you want to clean. And in the agriculture part, what we suppress is the pathogens in the soil or the biofilm in the pipeline. Hope that answered your question.
Yes, it did. Thank you. I think this one also ties into that. For nutrient capture and subsequent availability, are nanobubbles more effective for trace micronutrients, for example, boron for corn, due to their minute size?
We haven't on corn, we haven't tested on corn. We tested on other plants, but the nutrient delivery is actually better with the nanobubble generator. So you saw those plants are actually grows faster and larger with the nanobubble irrigation. But yeah, we might try the corn in the future. Let's see how that works.
Okay. The next one, suggested unit operations to burst stable nanobubbles for cell lysing applications.
Come again?
The question was the suggested unit operations to burst stable nanobubbles. And the question is referring to cell lysing applications?
I don't think we have touched that application. But if you are interested in that, maybe you can. I have my email address in the chat, and also at the end of the slideshow, we can talk more about that.
Okay, thank you. One coming in. So volume-based techniques are normalized, qualitative, cumulative distributions, and number distributions provide quantitative concentration. Is that correct?
Yes, so laser diffraction is one of the techniques that was shown kind of on my first slide. That's volume. The distribution output is volume. So that does not give you a quantity. It's not a quantitative technique that allows you to track how many particles or bubbles that you have. And I keep on using the word bubble and particles interchangeably because it is very hard to decipher which one is which. It would not tell you unless you add a contrasting agent. So coming back to laser diffraction, yes, volume will be sort of a qualitative technique to suggest what the bubble concentration is, whereas number concentration, such as nanoparticle tracking, it would actually tell you how many bubbles, how many particles you have in a given volume.
Thank you, Julie. One additional question. We have quite a few rolling in, actually. That's great. Is it possible for use nanobubbles for membrane fouling control in NBRs without killing any microorganisms?
Of course, but depending on what kind of microbes you are growing inside of your NBR and what kind of gas you're using in the NBR. But it is doable to control the fouling on the surface.
Okay, thank you. Do you consider pump cavitations for nanobubble generation a hybrid of Venturi and swirl or distinctly different method?
Those are distinctly different methods, and I don't see any like necessity to combine them together because so I mean the principle are entirely different. So and each one of them can produce nanobubbles. So I don't think that's necessary to like do a combination kind of generator.
Okay, the next question. Have you measured OH radical formation because of nanobubbles?
You mean hydroxide radical?
Yes, sorry. That might have been a typo in that. Yep.
Yeah. Our tech team are actually doing the research about the hydroxide radical. I believe we can have some results soon.
Great, thank you. All right, the next one. In what mechanism does the nanobubbles clean surfaces?
Like I mentioned before, it's the reactive oxygen species and the hydroxide radical helps to do the disinfection because those are highly oxidative species, helps to mitigate those microbes on the surface. That's how it do the surface cleaning. Also, the oxygen itself in the water can also generate hydroxide radicals or the RS species. We just enrich the water with the oxygens, way beyond the solubility of oxygen, and make it more efficient.
Okay, we have time for some more questions here. We have quite a few still rolling in. So nanobubble using membrane has advantages, but in case you are using it for turbid water, for example, algae removal from a lake, do you have problem of membrane clogging? Do you need pretreatment?
Yeah, I mean, pre-treatment is needed, but mostly it's just to remove larger particles, probably a screen to remove larger particles, so those particles will not damage the membrane. I would say probably that's one of the disadvantage of the membrane because those membrane itself does not cannot be damaged to perform this treatment. But the falling of microbes, like smaller microbes, is not a problem because the design in there, the water flows in the membrane chamber can mitigate that. So yeah, during the operation we have either in the lake or other application with dirtier waters, there's no falling, significant falling happen.
I see, thank you. How do nanobubbles interact with contaminants, microorganisms, or other particulates in the environment?
I'll say that depends. Like we mentioned earlier, the pathogens or the microbes are some of them don't like oxygen, so we put more oxygens in there to suppress them. some of them are afraid of reactive oxygen species that kills the pathogens instantly. So yeah, it's basically since we are using nanobubble, in different gases in there. So it all depends on what kind of gas you put in there, and it will have different interactions. For example, what I mentioned before are all using oxygen nanobubble to get rid of pathogens or microbes. But if you want to do it more, I would say aggressively, you can use ozone nanobubbles. That will give you maximum effect, and we all know how ozone works.
Okay, this is just a follow-up to that. You may have answered it already, but how stable are nanobubbles in solution, and what factors influence their stability over time?
Okay, so if you're not using them, let's say if you're not using that to disinfect things or grow algae, if you just generate the nanobubble in water and let the bubble stay there, it may stay there for weeks. It's very stable, stay there for weeks. Let's say 3 to 4 weeks. We have those detections, those analysis. So yeah, I'll say at least 3 to 4 weeks if you're not using. But the things may affect their stability is such as temperature. If you give it high temperature, it may go out, and if you give it very dramatic shake or I'll say stir, it may also get some of these bubbles out. But yeah, I would say basically they are very stable.
Okay, thank you. Just a few more questions here. How do you quantify hydroxyl radicals and other ROS? For example, what method do you use, and what concentrations of ROS have you observed?
Yeah, right now, like I mentioned, we are performing the research on the hydroxide. We prove there are hydroxide radicals and ROS species in there, but the quantified studies and more of those studies are still ongoing.
Okay, thank you for that. We'll do one more question here. Can you elaborate more about application of nanobubbles in drug delivery for some therapeutic options?
Yeah, this one we're not really even deeply engaged in there, but theoretically, since nanobubble are negative charged, so if you want to do some drug delivery, you might use this kind of feature to let's say attach the particles, the compound you want on the nanobubble surface, and deliver those bubbles with the liquid or other of the solutions to the place you want to go.
Okay, perfect. Thank you for that clarification. With that, we do have time for one more question. We're very interested topic here. Somebody mentions we are using nanobubbles to enhance algal growth. I have been using the nanobubble water to produce the nutrient with excellent results. Would there be a concern of killing the algae by inserting the nanobubble generator in the algal culture directly?
I'll need to ask what kind of nanobubble you're using here. Is that CO2 nanobubble or air?
He did not specify, but
If that's
he'll follow up.
Because we have already tried CO2 to cultivate algae, and it doesn't seem killing algae. Actually, on the contrary, the algae grow very good with those CO2 nanobubbles. But if you use other like air or other gas, I cannot guarantee.
Oh, he did mention ambient air.
Ambient air. There's not much of the carbon dioxide in the ambient air, so probably the the growth of the algae may not be as good as CO2, and but I think apart from that, it should be okay. Yeah, as long as you are not using some strong gas such as ozone kind of or pure oxygen, I think that'll be okay.
Okay, thank you for that again. with that, I think we'll bring this to a close. If anybody does have any follow-up questions, you can always email us at labinfo@HORIBA or reach out to Dr. Hua at PureNanoTech. Just a reminder that this is part 1 of a 3-part series discussing nanobubbles, so make sure to look out for our follow-up invitation to part 2 in June, and we look forward to seeing you there. Thanks, everyone.